Good afternoon. My name is Sarah Brodnick, and I am the CORS facilitator for the Office of Campus Research CORS at the University of Wisconsin-Madison. Please feel free to stop by our poster during the poster session to learn more about what we do. Next, I would like to introduce Dr. Steve Boppart. Originally from Illinois, Dr. Boppart started his career developing national laser safety standards at the Air Force Research Laboratory in San Antonio, Texas. From there, he went on to attain a joint MD PhD from MIT and Harvard Medical School. He currently serves as director of the Beckman Institute for Advanced Science and Technology and the Center for Label-Free Imaging and Multiscale Biophotonics, or CLIME, at the University of Illinois at Urbana-Champaign. Since joining the faculty at UIUC in 2000, he has published over 400 publications and has over 50 patents related to optical biomedical imaging technology. His research focus is biophotonics, where he has pioneered new optical imaging technologies in the fields of OCT, multiphoton, and computational imaging. Today, Dr. Boppart will present a talk entitled The True Colors of Cancer, Imaging New Biomarkers with Light, Not Labels.
Well, thank you very much for the introduction, and also thanks to Kevin and the organizers. It's really wonderful to be here, and this is the first time for me at this meeting, but already it's just such a strong sense of community and focus. So thank you for all of that. I want to touch on and start with some background, and from the clinical perspective, if we think about biomedical imaging, we tend to think about this imaging of scale, structure across multiple orders of scale. And similarly, if we think about the function that is so important to capture with imaging as well, we can see quickly this becomes complex. Now, from the clinical perspective, if we think about what modalities are used, those are the MRI, CT, PET, SPEC, really at this organ scale. But it is really light and biophotonics that allow us to be able to look at the cellular molecular scale. And this is truly where disease starts from that clinical perspective. Now, if we think about this as a surgical perspective, I want to give an example. One of the challenges that we face in surgery is that, for instance, the surgical treatment of breast cancer, lumpectomy specimens are removed, but we know that there is roughly a 25 to 30% chance of tumor cells being left behind. And it's simply because that surgeon doesn't have this microscopic view really of this level and scale of cells and tissues. And so if we think about how we might apply our microscopy techniques across these scales, but really for being able to look and intervene and give a microscopic view during surgery, then potentially this can have very real clinical applications. Okay, and if we do this, of course, not only is this benefiting patients worldwide, but economically and all the infrastructure can really be improved. Now, ultimately now, from the pathology perspective, what we're trying to do is optically biopsy tissue and shift from the pathology lab looking at the microscopic assessment of tissue to maybe being able to do this even intraoperatively in real time, and be able to capture even dynamics of what cells and activity and function is happening within the tissue. So it's all about changing the paradigm for when, where, and how we assess tissue microscopically. Okay, well, we're taking a label-free approach, and we had a wonderful talk about all the technology and developments of labels and how that is so important for giving that specificity. But there's many advantages that can come from a label-free approach, particularly if we think about this from a clinical perspective. Well, we want to try to extract as much endogenous intrinsic signal as we can. This can be then used to image fresh tissue, even in vivo. There's new forms of contrast that can be generated. And ultimately, we're not introducing any confounding factors about whether a dye or a probe made it to its target or did it land off target. If we're trying to translate these technologies into clinical use rapidly, a label-free approach avoids that FDA approval of a drug or a dye. And along the way, we have this ability to look at multiple physics and mechanisms that generate this type of contrast. Now, there's disadvantages, of course, to these label-free approaches. Like I mentioned, often we have limited sources of contrast, and that contrast can be, well, the specificity for the contrast that we're identifying can be low. But there's ways of being able to augment this with a lot of the AI approaches. So ultimately, what we're talking about and thinking about is, can we actually, instead of removing the tissue and adding dyes and stains to provide that contrast. And looking at that under a white light microscope, can we actually shape and tailor the light that we use to illuminate the tissue with, and then explore that wealth of optical intrinsic signatures that can come from that. So one of the label-free technologies that we've developed for clinical use and applications is optical coherence tomography, or OCT, based on scattering and reflections, simply the optical analog to ultrasound. But this is an intraoperative probe system we built that was used in breast cancer surgeries. And with a probe, the surgeon is passing this probe over essentially the margin of the tissue that was removed from surgery. And it was really about trying to identify whether or not there's positive margins. So being able to look at the cut surgical margin and see if there was any tumor cells evident or present at that cut margin. Well, and then of course, comparing this with pathology post-op. And we were able to get sensitivity and specificities in the range of the high 90s with this technique, and be able to rapidly scan OCT probe across multiple areas and large areas of that tumor margin, even the in vivo resection bed. Also associated with surgery, breast surgery, is whether or not the tumor spread to lymph nodes, and being able to use OCT in a label-free way to look at these lymph nodes, even in situ, and be able to identify structures and whether or not there's metastatic involvement. And I won't go into more detail here, but these are it's one example of a label-free technique that you'll see later will help us address this multi-scale problem. Ultimately, what we're focused on, the gold standard, is histopathology. And we've heard examples already at this conference of how this is an archaic technology, right? It's been around for more than 100 years, and essentially what we're trying to do is to move and shift that point at which we assess that tissue. In this case, these are just some examples of a negative and a positive margin using OCT. But we naturally asked, what can we see? What more can we see? In addition to just these margins, can we look at that microenvironment and what else is going on? Because maybe you admit that there's really not as much information in here as we think we could acquire and capture. So we're really taking on histopathology. And again, a very slow, time and labor intensive, very subjective process, only possible on ex vivo specimens. But in the end, it's an image that's really produced and analyzed. And so if we can generate those images in new ways, then maybe we can bypass that entire process. And in fact, can we actually begin to do this then in real time in the living tissues? All right. Well, like I said, what we're trying to do is essentially program the light and change the light, shape the light, create new forms of that light to excite the tissue with, to collect different contrasts and images that we can use for diagnostic purposes. So in the case, instead of waiting 12 to 24 hours to get that H&E stain section, can we then begin to see new forms of contrast by simply tailoring that light stimulus? So these are all images of that same tissue, fresh tissue, unstained tissue section or specimen, but with different forms of this tailored light. Okay. So collectively, we are able to get images that look like this if we put all those forms of contrast together. And I'll go through a number of these, but you can see that second harmonic, third harmonic, cars, intrinsic 2 and 3 photon autofluorescence gives us a picture of the fresh tissue, unstained tissue that looks like this. And I would say and argue that there's enough structural, functional, molecular, metabolic information in this tissue that we can get in real time that's valuable. Well, as I said, we really are tailoring the light. We generate a supercontinuum through pumping a photonic crystal fiber to generate a supercontinuum that's widely coherent, which means that these optical pulses are highly compressible down to 6 femtoseconds or so. And something short in time means broad in bandwidth. We have a large number of wavelengths that we can then select and use to excite all of these nonlinear properties of tissue. So simultaneously, with a single pulse, we can excite all of these nonlinear processes in tissue, and we can generate and collect simultaneously signals from each of these channels. And as you might imagine, the third harmonic is really going after interfaces and structural differences. The 2 and 3 photon are exciting autofluorophores, FAD, NADPH, related to metabolism, second harmonic for collagen, and then of course CARS or SRS for proteins and lipids. And we're really fortunate that nature has given us this palette of intrinsic signals that we can capture and put together in a highly multiplex but label-free way. We call this SLAM microscopy, which is short for simultaneous, label-free, autofluorescent, multi-harmonic microscopy. So quite a mouthful, but this ability to then, in a single shot, capture all of these channels. We get images like this. This is in vivo, again, unstained, label-free, fresh tissue, a rat mammary tumor. And here I just want to point out that if we zoom in, we see different colors in different cells. The tumor cells are these magenta ones up here. Well, we see fibroblasts that are either blue or yellow. That really corresponds to their metabolic activity and that change in color. But we can see collagen in green. We can see endothelial cells. This is one of my favorite images. It's a neurovascular bundle. And so this large vessel here is a vein. There's an artery that's adjacent. The magenta is really coming from the myelin that's wrapped around these in this nerve here. And these are adipocytes surrounded then by a collagen and fibroblasts that are generating that. Now this is in vivo, but it was actually a mosaic of multiple images, so we don't see the flowing blood in this case. But if we look at a single frame, we can actually capture longitudinal time-lapse data of the cellular activity that's happening in this rat mammary tumor. So we can see the blood flowing. We can see there's actually cells, white blood cells, intravasating, extravasating, in and out of the vessels. We see the tumor cells are accumulating, clustering around this particular area here. But you're also seeing we're tracking, and I've heard a lot of exciting results from this conference about dynamics and tracking, and this is again an opportunity really to apply a lot of this type of processing to better understand and characterize cells and conditions in tissue by their dynamics instead of just simply looking at structure or molecular composition on these H&E slides. So we've spent a lot of time trying to correlate our label-free multimodal images with standard H&E or immunohistochemistry, and that's sometimes hard because we're seeing structures and features that don't appear in standard histology or get washed out. And I'll talk about one of those, including extracellular vesicles. But the fact that we can see structures that really don't appear in standard histology gives us this ripe opportunity for new biomarkers and to be able to identify those such as extracellular vesicles. Okay, in addition, this is our form of label-free spatial biology or spatial omics. And what we can do is with this multimodal imaging and characterization, we can go in and characterize and segment all the different types of cells that are present and then begin to look at their associations with one another and be able to find, what cells and biomarkers are present and really how those are distributed. So we can go in and segment those, identify just a relatively crude subset of cell types, but then it just gives us this association spatially where those are located. And from that, we can look at different co-occurrence type matrices. We can look at just percentages of different cells relative in cancer versus control. We can look at, this is a metabolic ratio or the optical redox ratio, and we can see differences between cancer and normal. So many, many new label-free metrics that we can use to differentiate these tissues. Okay, we took this technology out of the lab because we really want to bring this to the point of procedure, and we were able to put all that optics into a cart. Underneath this little black box is a glass window. So in the operating room, the lumpectomy specimen will be passed to us or to my students, placed on that window, covered up, and then we can scan and collect these label-free images of that fresh, unstained tissue before it goes off to pathology. And we get images like this. This is normal human breast tissue. There's a lot of adipocytes here. Green, again, is collagen. Yellow is elastin. Another example of one that's a lot of normal collagen and elastin, but compare the organization of collagen there to the tumor tissue, where that collagen is highly disorganized. There's these blue and purple tumor cells that are now infiltrative and everywhere throughout this tissue. And this is ductal carcinoma in situ, so it's a pre-malignant state. This is a small little duct here, and you can kind of see the the collagen organization, the tax grades and organization around this particular structure. My students like to call this the tumor galaxy, because if you didn't know what scale you were looking at, right, you might think this came from a space telescope or not, but we're really looking at hundreds of microns in terms of these features. So hopefully you appreciate, and also coming from this meeting, that there's just an opportunity here to apply AI deep learning for looking at these highly dimensional spatial and temporal information and images. So that's exactly what we did. We developed this DNN to essentially replace this process. Instead of the tissue coming out and being sent for H&E processing, reviewed by multiple pathologists, coming to a diagnosis, which also only 75% of breast cancer pathologists will actually agree after looking at those tissues. But instead, really take that tissue out with minimal processing, collect those images, pass this through our DNN, and really get a reliable, consistent type of a diagnosis and assessment. And with this, of course, be able to reduce that delay and a lot of the potential angst from patients and their families with having to wait or have reoperations. So with all of those multimodal images that I was showing you, we collected these from 14 cancer subjects, 7 healthy subjects. These were women undergoing breast reduction surgeries, but no history of cancer, and collected images from multiple sites on those specimens, divided them up into training, validation, test set. We had over 50,000 tiles that we used in this particular model. And in the end, we're getting per tile accuracies of 95% and larger slide images accuracies up to 100%. So very good approach here. And essentially what we can do is to feed in any of these particular images, and then you can see how individual tiles are essentially assessed this way as being positive or negative for cancer. The unique thing about a lot of these approaches is that we can look at these t-SNE maps, and in this case, we don't have points. We have images associated with each of these, and we can look at what each of these images in these different regions are associated with. And if we look at those images that were associated with true negatives of cancer, normal cases, it was really the collagen organization in those images. The false positives, false negatives were really from these images with these elastin fibers and some of these other strange looking objects that we were seeing. But the true positive images for cancer were those that contained these extracellular vesicles. And this really just reinforced this concept of this being a new biomarker, label-free biomarker for this imaging. This was one of the first images we captured of tissue with our SLAM technology. And we were amazed by what all these little blue and white dots were. Nobody had really seen these before. And they ended up being these extracellular vesicles that have a high NADPH count within them. And given their size, they also have a very strong multimodal signature. And it was this ability to be able to see these label-free and know their spatial distribution, which really has opened up a large area of investigation for us and for many other groups. The standard way of assessing these vesicles is to take out the tissue, dissociate it, isolate, ultracentrifuge, and you completely lose that spatial orientation of where those vesicles are. These vesicles are very interesting because they are responsible for intercellular communication, but when they bud off, they really take with them a bit of the parent cell's cytoplasm, and that cytoplasmic milieu. And whether that be a cancer cell that's highly metastatic and metabolic, that same signature will be carried with that extracellular vesicle. So that's what a lot of the foundation of what we've been pursuing. Now, if we isolate those, put them on a cover slip, and we look at them with our multimodal approach, they only show up as a pixel because they're sub-resolution. These are on the order of tens of or hundreds of nanometers, but they have a multimodal signature. And if we look at that metabolic signature, we can see that vesicles coming from cancer cells that are highly invasive and aggressive compared to normal, we see a difference in their histogram of this metabolic optical redox ratio. And so it's essentially saying that, again, we can tell how metabolically active that cell was based on the signature from that vesicle, no matter where it is coming from in that body. So we went back to our clinical cart, and if we just simply count the number of vesicles in our images, there's a statistically significant difference between cancer and normal cases. We can actually see that those numbers go up as the more aggressive or the higher grade the tumor is. And we've been looking to see how these vesicles distribute throughout the body and get carried through. So a lot of further work trying to investigate how those can be used as clinical biomarkers for cancer. Now, really interestingly, if we go back to the human cancer images and the normal cases, and we look at these vesicles and their histograms, we can see it follows the same trend. The more aggressive the cancer, really this histogram shifts to lower values. But it was interesting to see that on 2 breast cancer survivors that we examined their vesicles, we found that even though they were clinically free of cancer, diagnostically, we found that their histograms, their vesicles, showed a signature similar to a very aggressive disease. And we don't know if that's because they had underlying aggressive disease or our hypothesis is that once a patient has a cancer, their body becomes preconditioned by these vesicles to host metastatic tumor cells or other cancers. And is this truly why patients that survive cancer are always at a greater risk for secondary malignancies over the general population? But it's something very fundamental that we're furthering to investigate with this technology. And this is just reiterating that point. These tumor cells send out these vesicles into the circulation. They get taken up throughout the body. These cells undergo changes in their metabolism and mass production. Our body becomes preconditioned. And do we remain preconditioned for long periods of time, even though that primary cancer may have been treated? So more to come on that, but something clearly to think about changing this paradigm. Well, the future really of this technology, we're just so excited that we've only begun to look at subsets of tissues. There's many other tissues, there's contrasts and things that we're seeing here that you don't see on histology in any form. And there's a lot more dimensions for us to explore. In addition, I think that this concept of a living biopsy is fundamentally new. So instead of that slide with that fixed and stained histology section, could we have a living slide that has a fresh tissue that's being cultured, right? And maybe preserved and maintained for 24 or 48 hours, where we can interrogate all these other metabolic parameters and changes before you kill it and do H&E. So, as a physician, I'm just really frustrated by all the efforts we go through to get a biopsy, say a liver biopsy or a lung biopsy, and we spend hours to retrieve that tissue. The first thing we do when we take it out is we plunk it in formalin and we kill it, right? And we kill it to preserve the structure and morphology, but we're really just losing so much more information. Okay, just to end, how this is all coming together, we were fortunate to receive an ARPA-H grant that is to develop this margin diagnostics platform, Margin DX. And it's this idea to combine OCT and these nonlinear imaging technologies into a platform to solve this problem for surgery. It's a large multi-institutional project that involves Mayo Clinic, Illinois as the lead, the National Center for Supercomputing Applications, and a number of other partners that are coming together to really solve this critical problem that ARPA-H has identified. And it's essentially this combination of OCT for the fast, large, wide-scale imaging, followed by nonlinear imaging for this high-resolution digital histopathology, coupled with AI on both of these technologies. This solves that multi-scale problem, because how else might you microscopically image or survey, tens of square centimeters of tissue in these particular examples? So with OCT and AI to scan rapidly, identify regions of interest, to be able to then switch to a robotic arm that could help stabilize and do the nonlinear imaging during surgery. And we really think that this is going to change the surgical workflow and eliminate any of those positive margins and the need for patients to come back for repeat surgical excisions. And then finally, this technology can also be applied for surgical robotic procedures or laparoscopic procedures as well. I just want to thank my research group. There's just been a great interdisciplinary team of people and collaborators that come together for this type of work. Many of our funding supporters and agencies across different domains to support this interdisciplinary research. And one in particular, support for our national NIH, NIBIB P41 Center called CLIMB, the Center for Label-Free Imaging and Multi-Scale Biophotonics. And similarly, this is a P41 center that is to develop these label-free technologies and share them with the community. And that's exactly what we've been doing. I was really thrilled this afternoon to hear so many of the different courses and microscopy courses and opportunities. I know we might have a student and postdoc and scientist audience here. So I want to point out, as part of our center, we have a summer school, 2-week summer school, where we cover the principles of biophotonics, but also hands-on labs, industry contacts, a lot of industry representatives that talk about different career and development paths as well. So if you're interested in this, please feel free to reach out, and you can see the contact and website information as well. I just want to thank all of you, and it's been a wonderful conference so far, and happy to answer questions.
I think we have time for just 1 or 2 questions. And there's a comment online about getting in touch with Anoscope about mid-body remnants during cell mitosis, and we'll share that with you later on.
Okay.
A beautiful talk. I was just really struck that, you're well on your way towards the dynamic fingerprints, and in addition to multimodal, have you looked at getting more out of fluorescence? Like in addition, I know you're working on FLIM as well, but like looking at polarization, rotational energy, the spectral shifts.
Yeah.
Seems like a lot more you could add to that.
So absolutely. I think that there's so many other intrinsic signals available here. So I mentioned a lot of the fluorescence, just intensity, but we also are capturing lifetime for those. Our SLAM system is also expanding to 4-photon excitation of tryptophan. Again, because we're dealing with such short optical pulses that nonlinear signal generation is so highly efficient. We can get a strong 4-photon signal there as well. But you're right, polarization, all of these opportunities to expand. Our SLAM platform just involved 2 and 3 photon, second and third harmonic, but there's CARS and there's other, SRS and others that we can implement. A lot of what my lab's doing is just building these multimodal systems. And in a sense, I think, again, more is better, that the more dimensions we have here, the more precisely we're able to define kind of the state of the cell or that tissue, and whether that state be healthy or disease. So.
Hi, fantastic talk. I'm here.
Okay.
Yeah, fantastic talk. Have you been able to explore the relationship between metastatic cells with the EVs in your tissues?
Yeah, so that relationship. So I think this hypothesis that we're developing here is that these EVs are produced by those tumor cells well before they're metastatic. And that changes this thinking. We think that, the tumor begins to grow, it becomes a large enough mass, cells break off and they metastasize, right? But what we're seeing here is that these early tumor cells already begin to increase their extracellular vesicle production. Those start circulating. They're appearing in the periphery in other cells. And we have a study where we looked at this co-occurrence of these extracellular vesicles in the periphery with cells that have undergone metabolic and mass production acceleration in the periphery, normal cells. And that's what's leading to this hypothesis that there's preconditioning. And so then what happens is when those metastatic cells do arrive, they land in that, that soil hypothesis, that the soil has already been fertilized, right? And they're there to then grow. So if that's the case, right, then we have to rethink. It's not, we've always heard metastases are what kills you, right? But we have to stop and think about therapeutics that stop that extracellular signaling that happens very early. So new challenges.
Hopefully this will be a quick one, Kristen.
I was wondering if you could speak on the imaging depth that you foresee being able to get when you're using these kinds of imaging modalities, like during surgery. And then also, if you could speak on the kind of learning that you're using or network that you're using for the AI portion, like is it supervised, self-learning, that kind of thing? And then how you're validating that they're actually calling things correctly.
Yeah, yeah. So I think if I captured that right. So some of the examples I gave for the cell segmentation, that was really done under some supervised learning, being able to work with a pathologist, identify cell types on the histology that matched with our images. And so we were able to do, that was primarily, that type of directed type of association. For the SLAM imaging of the breast images, that was done through kind of weekly supervised learning. And I didn't show this, but the thought was we're never going to have enough of these perfectly co-registered label-free images and corresponding histology, especially if we're looking at cell-to-cell level, right? And so, and in many of these AI models need hundreds of thousands of those pairs, and we're never going to get that. It's a common problem. But so instead, what we did was create this weekly supervised approach where we just created these bags. We knew that this tissue came from the core of a tumor, right, in this patient. And then we knew that these tissues came from the periphery of that tumor that the surgeon said and the pathologist said looked normal, okay? And then we had this bag of samples that were just from completely normal patients. And just by creating those three, we let the algorithm identify what were those patterns or signatures that created found within those tissues in those higher level bags. And in fact, from that, we were able to identify which modalities gave us the best diagnostic utility. That collagen signature came out. But that weekly supervised approach actually pointed again to those extracellular vesicles, and that those were the new biomarker that was discovered. And I think setting up these weekly supervised approaches that way opens us up to identifying potential new biomarkers, because you start to see clusters on your t-SNE maps that really correspond to something that might be completely new. And that's what we were seeing here.